Yes. তুমি বলে দিও ওদেরকে কোশ্চেন এখানে পাওয়া যায়। Hello, welcome everyone. This is our second discussion session. For today's discussion, Shoma will help me to conduct this discussion session with Akash. Akash did his BSc in agriculture from Anushandhan, Shikha Anushandhan, and did his MSc from Ramakrishna Mission jointly from the ERI, Philippines. He is a bright student, and I'm glad he came forward to help us to collect all questions and your suggestion from the YouTube live chat box. Today my concern is we are discussing a section which is very important, but it is very difficult to cover up within one and a half hour maximum. So I try my best, but I would suggest if you guys feel that it need more and more, then I ask you after the end of my talk, if you got the interesting, then I think that we can go a sequel of photos related to manuscript and photo related things. So yesterday I'm thinking that I should break it into 2 or 3 parts, like part 1, part 2, part 3, in different time interval, because my plan was I just go for the demonstration. Then I realized that when you know the technique, and I'm sure for everybody knows the Photoshop and this all. I am not the expert of these things. I'm a biotechnologist, but I do think that we need to learn some basic principles, because nowadays it's available. If you have the money on your project, or you wanted to wanted to help any professional help for your photo editing and your style, then it's not a big deal. Many companies are there to help you, but you have to know about something which maybe, because you can't get the help for everything. It possible for one to high impact journal you can do for the you can do by the expert, but for everything, like for MSc dissertation, for the lab presentation, for lab work proposals, and everything you can do by purchasing. It's very difficult for anybody. It's not for developed country or developing country. No one wants to give money without any requirement. So my suggestion is that I learn these things, and I'm very interested in this field because it's lots of others thing when come to the our life, researcher life. So uh, I I am very interested in this topic, how to write, how to edit, how to communicate. So it's it's one of my key interest to photos, because it's everything. When we go for a paper, we first see the photos, then we go for the other things. Okay, so I see the photo first before going to the name, country or author's name. I just go for the photos first, then came to this thing, then the obviously then the abstract, then the introduction, result, conclusion. But photo is very important, and many of you maybe not know, sometime you did a mistake which is a crime, like a crime in today's editorial board or editorial system, because maximum editorial managing softwares are now AI based. So if you change anything in a photo, it's look nice to you and your lab mates. No one can find the mistakes, but not really. It's not so likewise. Why it's not really happening? Because nowadays everything is judged by the computers, and it's a little bit tricky. You can't do any manipulation in the photo, and it should not. So by some mistake you can do, if you have any such in such a query, you can post it in the live YouTube chat. Shoma and Akash will collect the questions and I will ask you. So let's see, I start with some basics about the photos and their representation. I say it's a visual communications. It's a visual communication thing. Then we go for the little bit Photoshop and how to manage like PowerPoint, which is the most commonly available softwares. But I'm not expert of these things. I'm not the graphic designer, or I am not the animator. So I'm a researcher, and I follow these principles and I try to maintain these things. So I wanted to share with you, because this discussion section is for this purpose. If anyone has any better suggestion, they can ask me. It's very open to, and it should be, because it's a discussion. You have to ask so many questions. We try to discuss, but due to limitation we can't afford so many people in Zoom. That's why I'm go for the YouTube live. But if you wanted to share and you come into Zoom as a panelist, you inform me that you know Illustrator, or may you know this type of things, which we can incorporate it in subsequent series of discussion. And very beginning, Shoma will tell you guys, today we don't get any certificate, because it's a practice session. It's not a certificate like thing. So I just wanted to clear in very beginning. Okay, Shoma, just introduce Bioengine.
Hello, welcome to the Bioengine.com. We have started a new series, Bioingene LIVE Discussions. It is mostly a peer-to-peer discussion section dedicated to research in the field of life sciences. This new series is a part of the Bioengine initiative. It is absolutely essential to keep trying to improve the Bioengine platform by incorporating initiatives that would be helpful for researchers and research enthusiasts. Our webinar topics and expertise of our speakers are an integral part of this initiative. Apart from webinars, we are now adding some extra effort to make a scientific community for discussing important aspects of our research life, like research planning, conceptualizing, methodology designing, manuscript writing, finding appropriate journals for publication, communication research and publishing, and of course, like for today's topic, proper figure preparation for your manuscripts. The bioengine.com live discussions is such a platform where you can address and discuss such topics. I invite you to join this initiative like all other Bioengine initiatives. This is also a free for all and dedicated to our young viewers and participants who want to actively get involved in this discussion session. This series is purely for for the development of knowledge and awareness on important issues related to science and academia. If you are interested and want to participate in these discussions, please visit our website and register in the discussion section. You can share the topics you want to discuss, which would be helpful for researchers and future scientists. And of course, I would like to add that this, as Shubho said, that this section is only for knowledge and no certificates. Our topic for today is how to follow journal guidelines on figure preparation for your research manuscript, live demonstration using Photoshop and MS PowerPoint by Dr. SHUVOBRATA Majumdar. Dr. Majumdar is a DBT Postdoctoral Research Associate at the University of Calcutta, India. He completed his BSc Honours and MSc in Biotechnology. He is a molecular breeder of fiber crops and a trained professional in rice research from the Erie, Philippines. He is also the founder of this platform, Bioengine. I now ask Dr. Majumdar to please share his screen and begin the discussion section.
Thank you, Sean. Sorry, I wasn't made. For today's talk, I divided it in a three section. First, I cover a few things, few basic bio presentation. Then we go for the short practice or demonstration by the softwares like Photoshop and all, and then go for the question answer round. So for the question answer, you guys can ask the question or any related or non-related. Sometimes you can think that it is talk related or not, but not in discussion, don't worry. We try to focus this format in webinar. For discussion, you can ask anything, any questions related to that. Okay, so I start the screen sharing. So it's a very common and famous quote, a picture worth 1,000 words. And why it is important when you write a manuscript or report, it attracts the audience. Your picture can attract them by the quality, by the appearance, by the style, or they may found it's boring. If you have many pictures in your manuscript or paper, but these are not so good. So for doing that, as I say, I'm not the expert of in this field, but I like to acknowledge one person, uh, that Luke Cox. Dr. Luke Cox is a founder of Somersault 1824. It's a company in the Belgium. I learned many things from his webinar talk, and I today I just discussed this experience from the given by the Dr. Lucas, and I try to focus some key components, uh, and as I say, we need too many examples. So I don't wanted to hurt any scientist, any researchers, but here I took some examples for better understanding. And that is the reason I acknowledge more Dr. Luke Cox, because these examples are taken by him. I'm just presenting it. I don't want it to hurt anybody, because sometimes it's not very difficult to follow all the rules in the manuscript. So first, where they're very important, where we go for the visual communication, where the visual communication is more applicable. In a manuscript, graphical abstract is a key component. You can design a abstract because graphical abstracts is available freely when your journal is under subscription. If it is not an open access journal, then viewer can access everything. They can access only the graphical abstract part or presentation slides. It's very important for professionals who are selling the products, like any products, car or anything, cosmetics, any food products, or any commercial products. It's it's very hard to applicable or use the visual communication in experimental reporting on graphs. After the viewing this discussion, don't try these things in the graphs, because it's a scientific and it's not require any such things for the graphs and experimental reporting. So it applicable in few area, because your manuscript is combination of all. You have a graph, you have the pie chart, you have the line diagram, and also you have the few pictures. For me, there are many pictures of insects, many pictures of plants, many pictures of their seeds. So I use this knowledge when I took the picture, and I also use these things when I edited for the manuscript preparation. So there are few components like line, space, shape, color, texture, size, positions. These are the very important for visuality perspectives. Like I'm sure what is written here. Many people go for the you don't matter, give up. Okay, because it's our instinct, we follow in this way, but the presenter give us gap here between the two things to describe that you should go for you matter, don't give up. Okay, so it's change the perspectives, the meaning, the what the actual message. And so we have to you have to very conscious that no one go for you don't matter, give up. We should assure that everybody goes in a way that you matter, don't give up. Okay, then how to do that? So it's a rule. I'm sure everybody go for the word compose. Okay, you not go for the bottom right or any others. You not go for this section. You are not going for this. You first all of you maximum are go for the compose from the top and right, because it's our technique, or you can say that our brain developed in such a way, we go for that way in that. Then there are 3 arrows. Then we go for the high density or deep band, deep band or deep color arrow, which straight forward to the right, that follow reader's eye over, because it's more thicker than these two. Then you have to decide that where you wanted to go, where you want your viewers or your readers to see the matter. So sometimes it's arrow is very acceptable and appreciated method by many journals, but you can do any other way. I come at later. So it's a basic thing. We start from here, then you have to decide where we should go from this side, or this side. But generally, we are looking for in that way. It is called F fashion. So in design, these 6 things are the key players. Maybe there are more, but these 6 things are very, very important: balance, proximity, alignment, repetition, contrast, and white space. Okay, so I go for the first, balance. Balance, it's not a physical balance. Okay, it's a visual balance. So visual balance can develop by few things. I just cover it. This is a unbalanced picture. Everybody now, I'm sure, if you have a feeling of anxiousness, or it's not right, because in one side is a very high load and one side is very low, so it's not acceptable visually. Maybe it's not a what I say. It's a presentation, but we should remember, because it create our visual tension. Okay, so how we can create this picture is better, much better, because your tension is now lower down, because here is something is a balancing. Okay, so it's called the balance, and it's the visual balance. Okay, so it's depends on so these are the visual balance. You can create the weight, you can create by size, color, value, position, texture, shape, and orientation. Like red is heavy, yellow is light, black is heavy, gray is light. Or the size and large size or small element, you can create a balance by these things: size, color, value, position, texture, shape, and orientation. There are some basic style like symmetrical balance, asymmetrical balance, and radical balance, because we are use these things in our research. If someone write a review paper, then maybe a few review paper on CRISPR, then if someone can use here the CRISPR and here the agriculture, medicinal, and others applications. So it's a style. It's a style of representation. It's called the radical balance. Or sometimes you can balance in such a way, like when is a control and test samples are there, we can put in such a way, or we can try in this way, but preferably symmetrical balance is good and acceptable. So it's an unbalanced figure. Okay, there are high load in one side. Okay? It is not a unbalanced figure. One example is that this this is taken from a paper published by Elsevier. There is unbalanced, because it's look like okay, but it's not visually correct. it should be in such a way, like this one first, then arrow, then this one, then that. Or if you have no space or you have specific guideline, then you come from top to bottom. But it is not a right thing, because viewers searching for these arrow, because it's create a situation that you need to go in that way. But it is not a arrow thing, it's a dead end. So it is not a good representation, it's a unbalanced figure. Then proximity is a related things, because now I'm right now I'm not seeing the YouTube chat, but I like to you guys to enter the answer of this thing, that uh, here sorry, I forget I create the animation. There is a 16 dots. Okay, you can count 1 2 3 4 5 6 7 8 9 10 11 12 13 14 15 16. But it's very difficult to understand the 16 dots here. You have to calculate, you have to count. But don't do in such a way in your manuscript, because lots of paper are published per day. No one have the time to do this type of things. But you can do in that way. You can group or club. It's called proximity. It is very easily visualized that there are 16 objects. So it is also a proximity issue. It has to test, and it maybe it creates some results, like fever or cough, dressing, this type of things. So it is limited proximity. Okay, it could be better if it can be corrected in such a way that these are the cause and these are the effects. So the corrected orientation, it will be better if it in that way, but it's not a good representation, though it is published. Don't It's published, and it's published accepted journal, but it is your duty how you wanted to feel about your viewers or your readers. Now alignment. The alignment is a where you put your things. Okay, there is a two alignment. This top one is a poor, and this one the organized. Why it is poor? Because this text is a left aligned, and also there is a center aligned. But when you go for this alignment, there is a clear gap. It is optimized alignment. Everything create a white space here. It is important. I come to the white space section. It is better than this one. Like here, it's not. It is one type of alignment that you put in different way. It is center. It is left. So it would be better if it happened in that way. So these are the alignment. And for the alignment, we can use a technique that is called grid technique. Grid is very common. Grid available in Photoshop and any designing. So this is a simple grid. Sometimes people say that grid are for the novice or new people. Those are new for the software, but grid actually used by professionals. Okay, like this complex grid is very famous because Apple used this grid for their. This is the Apple photos. This icon is for the Apple photos icon. So they use this grid. So grid is a very good alignment tool. It can help very much. You should align your figure with the grades. You can on or off the grades if you have a problem by writing or anything, but you should check about ultimately on the grade that whether it is aligned or not. It's a famous logo, it's the logo of Apple, and it's has the grid. It's called golden circle. You can see that all are circles. If you see the logo, you can you can find that there are so many circles, but actually it is made by so many circles, and it's called the golden circles for the Apple logo. So it's also one type of grid. So everybody use grid for perfect alignment for a product. Like there are so many issues of alignment. Okay, it's a accepted paper. It's not that that's so bad, but there are issues of alignment. Like there are gap between these are different, boxes size are different, alignment is somehow different in the text also. It's left aligned, is a middle aligned, is a middle aligned, is a now so it's very difficult, and it should avoid this. It's not that it is not acceptable by the journal, but you should avoid this type of alignment. You try to align this. Another published photos that is very difficult to how to import is the mixture. There was no rule of that type of alignment, and you should avoid such type of drawing. Then repetition. Then repetitions is a designing things. Sometimes you can repeat, like when we go for the graphs, uh, say there are 3 samples, sample 1, sample 2, sample 3, and you did hype experiment on them. Then you should use a same color or same texture or a same tone for all individual samples. So it's create a smooth reading, or it create a conceptual presentations, because when our reader go for that color, they automatically know that this is for sample 3 or this is for the sample 2. So repetition is very important. Like here, there is no need that these 4 and these 1 is the same category or same product, because you create a pattern, the color pattern. Here is a 4 repetition, because here you use the color for the labeling, and you also use in same color, you use every here and also here. So it's not a good, it's randomly repetition. It should avoid it, though is the published figure. Contrast is a important thing. You need, and I go for the contrast editing in Photoshop, but it always add the visual interest, or it's very important. You can create the contrast. You establish the contrast by these things: is typeset, size, color, shape, line thickness, etc. Or there are so many others method also. But the contrast is very important. Like that, there is a high contrast. It because of the color is darker than the small one. Also for the size, there's a larger than the small one, and also for the text, it is the high size, higher size than that. So you clearly create a contrast. Sometimes people are intentionally do that. They can put this picture here and it is here, because we go for the F fashion, but create a contrast. It is also a rules that you put something in that way. That's why we try to put here control and here the text. This is another aspect, that sometimes you intentionally break the F fashion pattern of our looking to create the contrast. There is a very lack of contrast here. The color, you don't need to color this type of boxes, because I prefer, I am very habituated to seeing in students and schools and colleges, they color every boxes, but don't need to it, because it's create the contrast issue. It's very hard to read anything within that. So you avoid this type of contrast. Don't use the background color. Here you can see only the red. It's very hard to read within the boxes, but your intention to focus the boxes, not the background, but you use the wrong contrast. It's create the visual, it's not visually right, and it's create some tension too. Here you only see the arrows, but your intention is not to viewing the arrows. Your intention is to how products are developed, which byproducts are there, and how these methods are going. But you contrast so many, create a contrast and focus on the arrows, so you only see the arrows. Then very hard to find that, okay, there are some copper, cadmium, and these things. So it's not a good idea. This is one picture of my work. It is um, when you try to focus in this type of picture, is sometimes it's come to the background because it's a rice and it's live plants. I can't. It's not the harvested one. So it's very difficult because here the green background is create the problem, because also I wanted to focus those are the green grains. It's a crossing product. So it would be better if you place a black paper on that, then it create the contrast. It's easily keep your focusing element in the font and suppress the green color. So using that black uh, uh, sheet paper, either cloth or anything, it's very helpful because it actually creates the difference between the contrast. It's the purpose of contrast. White space also very important, because this picture is a published picture and is very uh, artistic in the way, but it's not correct for the scientific presentation, because here it's a full figure and something are written within that figure, so it's very hard to read. So because lack of white space, don't forget these are the white, but I'm not talking about the white color. I'm talking about the white space, which is in which should be introduced within the uh, photos during the preparation. So now because I already told it's very difficult to cover everything, so I'm focused few of things, and I did not cover the color and others important thing. Maybe in future, or I ask you guys that whether it's I should arrange one, then I can go for that. But because color is very important, but today I'm not going for the color. So arrows, there are two type of arrows. One is called open and one is closed. For manuscript, closed arrow are accepted. Don't go for the open arrows, okay? Because when you go for some author's guideline for reputed journals, you can find that they clearly mention to use closed arrow, not the open arrow. And the arrow size also matter, and it's sometimes varies between the journal to journal. Like ACS publications, no thinner than 0.5 not acceptable, where cell press accepted 0.35 to 1.5 points. So you also need to learn before starting on preparation your manuscript. That's why I prefer always one, because it's within the range of every journal. I think so. One, I prefer one, but it's also depend on the journal to journal and their guideline. Now the font. This is two type of things you have to know. One is a typeface and one is the font. Typeface is a category where the fonts are belongs. Sometimes you when you type, or I see, I just open my MS Word and I can demonstrate it. Some typeset has different fonts, and it's very important you choose such typeset, because it create your visual differences. It would help you to create the balance, to create the emphasis, to create the importance, and you don't need to the color to focus on, because you focus by the font. There are two categories, serif and sans serif. Serif are the font for the printed material, books, or any printed, offline printed papers, there this is the acceptable because of this notch or help to reader. But for the online or for the in silico or computers, it is the sans serif or this type of font where the notch are missing are the acceptable. Here I mentioned few font because it's very difficult which font I use, because when you start it's searching a font, there are 200 or thousands fonts are available depending upon your writing softwares, but you have to focus. Those are the very popular nowadays and more acceptable. Arial and Helvetica are the more common font for non-serif, sans serif font. You should use Helvetica or Arial, but it is your choice. There is just a suggestion, but not always, because when we go for the blast sequence or go for the nucleotide sequence, we always see this type of font. This is called the monospace font. Every letter actually need the same width or same size, and it is help to understand the length of, when you compare two DNA sequence, it automatically help you which one the shorter and which one the longer, because the font is a monospaced. So monospace form are only for the sequence queries like in BLAST or in multiple sequence alignment. Every others applicable when we use the RNA and DNA sequence, we should use the monospace. And by default, NCBI use the monospace. So this is my first part that I go for the presentation. Now I wanted to give you some idea about the editing purpose. Sorry, discussion. Then for the editing, you need few things, like you have to click your figure, because when you start the manuscript is the end of your process. Very beginning you start your experiment and doing the figures. So these all are raw figure. I'm not correcting anything, but I wanted to see that you have to uh, click in a right way and right equipment. Okay, because it's need the details. Okay, don't use your mobile camera, though it is so high now, 45 or 50 megapixel camera is available. Use a traditional camera, don't use the mobile camera, because otherwise you lost the details or detail of these photos. There are these are the few photo you can find the details, and when I zoom it, it's not breaking. So first quality is a depth image, otherwise it's very hard to any good publication, because you need a good quality and depth figures. And do everything like background, foreground, do start is very beginning, like it is a raw figure. I am not doing anything, but I'm using these figures to create a publication ready for a demonstration purpose. These are the yes, all are my plants and my related research related pictures, but these are not so important. That's why I'm showing it here. So here I already create a background, give a scale. It's very important when you took the figure. Obviously, you need the scale, and use same like similar size of pots also. Here, though it's a soil pot, so it's not mechanically created, or so it's handmade pot. It can vary few millimeters in every pot, but it acceptable because it's a size or very similar. And when I took these photos, I arranged these things, because you can do anything within the softwares. If you did anything related to that, you can put a scale, you can put the background, or it's not acceptable, and it's one type of falsify, or it's not right way. So click your figure very beginning in such a way, and obviously when I'm in MSc or early PhD, I didn't know everything, so I clicked many figures without the background, without the scale, and these all are not accepted. I deleted most of them. So you should go for, and this is the reason why we should discussion section is maybe some people may benefit it, that okay, we should take in such a way. This is also a figure. You can see that it's a rough figure. When I use for the manuscript, I can edit something, but I should not edit the scale, background, and anything. So that's why you have to click the figure very beginning in very systematic way. And I prefer use camera, not mobile camera. Okay. Why should you not use? Because they are these file are the raw file. Now I go for the how to create a uh, publication ready figures. Okay, so there are few softwares. Very common softwares is the uh, PowerPoint. We we all have the PowerPoint. We can go for the PowerPoint and create a figure for the publication. Okay, but there are few limitations. Like if I have this figure, this is the figure, original figure. You can crop. There is no limitation. You can do the cropping, no problem. But you should not avoid any information during the crop. Okay, crop is allowed, but you should not use it uh, to suppress something. Okay, so it's the crop. Then you put the name like control. IR 64, and use another box here, but I prefer use this same box. Why? Because it's maintain the line paste IR 64 6. So I use same box because when I go for any changes, it's helped me to keep the lining. Otherwise it's very hectic. You sometimes you create up here, down here, up here, down here. It's not a good process. It's a process. Then it is maintain one same font. Here I wanted to use the Arial. It should be within 12 to 14, but it's not very difficult here in case of PowerPoint. That's why I took little bit higher value. Then you can group this figure. You can group this both figure and save as picture. Generally, TIFF is the acceptable form of most of the paper. PNG and JPG also acceptable. You can save as JPG, and name is the PowerPoint test figure 1. Okay, so you don't have the control on the site. So this is our figure. Okay, same figure, same things. I wanted to edit in Photoshop. first I just go for the edit portion, then I can go for other thing. So in case of Photoshop, okay, it is a grid. You can remove the grid view. It's the show and grid. If you remove that grid is removed. But I suggest use the grid. Okay, so here I can crop once again like last one. You can go for the YouTube Photoshop. Many channels. I'm sure you can find the how to use Photoshop in the YouTube, and I'm sure they are more better than me. I'm not so expert. I can do my work just. Then here are options that you can use to brighten and contrast. Don't use so extensively. You can use, and you can use for the same way. You can change the brightness of that compared to that. You actually you change the brightness of whole picture. Okay, so I wanted to change the contrast. If I change it, it look more black background. Okay, and I lower down the brightness. So it's more soothing. So you can use this thing, but you don't change any locations, particularly any location. I change the for the whole picture. I change it for the whole picture. And second thing is the raw picture is always with me. If any reviewer or any editor asks me to give the raw picture, I can give it. So that is the reason that I little bit change the contrast and brightness. Then I can put, similarly I can put the name here, the test, and like I already I use the color. You can change any color. So it's the test, and obviously there are the control. Sorry, it's the control, and that other one is the test. Is a control IR 64 seats, and another is a test one. Test IR 64 seats. Why these takes are so small, though is the point size is so high? Because this picture is took from the DSLR, and the size of picture is very high. Actually, it's a very big size picture. I come to the next topic, that what should start, how to start. So it's a picture. Then you have a many option. Okay, to use the TIFF, which is more required option. Use the same things, but not JPG, is a TIFF file, and it's a PS Photoshop test file. You avoid the layers. It's create the size of your picture, and it's very difficult to communicate because it's a certain amount of size. Only compress use this one, not none, because it's create the size. But LZW actually accepted by the journal. So now we have a two picture. One is this one, another is this one. One is created by Photoshop, and another is created by PowerPoint. Okay, so definitely Photoshop gives some extra input on that. That's why everybody use the Photoshop. But Photoshop is a dangerous software. When you use the term I'm using Photoshop, it's very difficult to know what was your purpose, to avoid or to fabricate your picture, or just edit your picture. So Photoshop should be used in such a way, otherwise you can cut, because nowadays AI system is used for these purposes. Okay, then I should come here, but you can use any size of pictures. Okay, because already given by the instruction, by the manuscript, that your length of picture should be that or should be that. So before you should start, uh, or I just, there are a new option should start in that picture. Generally, one A4 is a 1/12 inch in the size. Resolution is the matter. If you change the resolution, lower down the resolution, size will lowering, and if you increase the resolution, size will increase. I prefer 300 resolution, which is default. So it's a picture of a A4. It's a size of a A4 sheet. Okay. So uh, when you start your editing, first you choose your size. Actually, which size you wanted to uh, present or publish or print out. Then you choose the you just then you collect the picture. And I ask, it's a large picture, so you need to adjust within the file. Okay. And alignment, you should, that is the purpose why you should use the grid to check that, okay, yes, it's okay, it's little bit same in the here and here, so it's perfectly aligned. Okay. um, and this picture, if you have another picture of anything where you can put, like I go for the, this is my file, I open another figure, like the rice. uh, okay, this one. Okay, what I can do, I can do the cropping, because these things are not needed, but I'm not crop in such a way, because if I crop here, then it lost the height, actual height. So I should avoid the cropping of any information. I wanted to repeat again, don't crop in such a way that your information is missing. It should not be. Okay, so it's my uh, it is the size. Now what I can change, I can change a little bit brightness and contrast to more bright. And don't use other option too. There are a lot of options to change the color, change anything. Don't use these things. Use Photoshop for very minimum purposes, to size, to actual size, to crop, brightness, contrast. Don't change other options. I'm sure you guys are know about the other options as well. So if I wanted to put this picture in a collage, and I already created this collage. Now the grid will help you, because if wanted to use the same size of, not same size, same distance. So grid help you to align in a same. This is the perfect, and it has to be. And when you use this, don't drag in that way. Always drag in this way. It's maintain the integrity. Otherwise, it's flattened, which is not actual. Okay, so size is match. Then in between space, it's also matter. So use a specific in between space. If you want, then I use this space. Okay, or I can in such a way I can, okay, this is and this is, so there is a clear same space between two picture. Then if need anything, you can put here another picture or another thing, but I just go for these two picture. Hi, obviously you can crop this section too because it's not needed, but I prefer to in that way to keep it in the middle of the page. Okay, so it's our picture. Then you need to the labeling. It is, you need the guideline because sometimes capital A is accepted, sometimes the small a is accepted. And for the black, there are a lot of black option. Use a specific black, and for Photoshop, use these things. Make it 0 for the black, and it's always same uh, intensity and same tone of black. Then it's 12, or you can put it the 14 for labeling, but here I put the 18 to visualize. Here's a capital A. There, you should check the whether it is capital A or small a. It's specific to journal, and always put your figure either this side or in the middle or not here bottom of the figure, but always prefer position the labeling in that location. Don't use in that location or otherwise your reviewer comment on it. So here I need another little bit gap, and here I also put the B. Okay. And check whether the B is the right position as A or not. It's also important. So use the grid to know that thing. It's not a right. So you use, okay. So it's an aligned picture. Now we can save it. Save as a figure 1 final desktop. Use what you want. You can use TIFF, PNG, JPG, whatever, but prefer the TIFF file. Remove the layer, then save the OK option. Okay, so you already have this figure. This is the final figure you can use for the publication purpose. But I wanted to say something like I can color the pots because it's not so attractive. It's algae within that, but don't do that. It increase your manipulation in the figure. So it's natural. It's not harm your data. Okay, but you can prefer, you can clean the pot before clicking the figure. You can clean, sometimes we clean if algae is more than, and it's very obvious in rice pot algae uh, commonly found, but you have to regulate it is another thing, it's not related to figure. You can color the background. These things, this ground, you can put here black also, but don't do that because it's uh, it's lost its originality. Okay, so it's the figure. Now you can see that as I told that it's a A4 size figure. So I can put this figure. No. Okay. So as you see that I create a A4 size figure, but it's not A4. It's not the fault of this figure. Actually, this figure is A4, but it is on because of my word and which page I used. So in publication, you need to exact size. Don't bother about this one. If you for the your internal review or for anything, you can use any type of structure like this to save your page or save your the cost of color printout. But this actual figure is A4, so you have to pre-decide that this figure is important, so I can put the whole page for it, like I can, okay, and you can do that during your proofreading also. No, I need the I need the whole page for this, or I need this type of uh, space, like this one. Like this is a whole page figure. I prepared this figure on that way, and here I use this one tone, because the plants are different, because there's are the different generation also. But I use that same tone, but I'm not covering the color for today's discussion. That's why I'm about it. And so it is a whole page figure. I created in A4 size pages, and I submitted when they are proofreading. I told them that I need a full page figure of it. Here also, I created is a full page, but they design in such a way, and it's okay. It's not need so many others. Another thing I wanted to say, that designing or concepts, like this figure. This figure, you can see the whole story. It's the different things, like it's the vector construct, it's the explant shoot tip, it's the selections, it's the growing of a bio plant under greenhouse and the bioassay, and this is the harvest of the plant and check the quality of fibers. I am working on jute, so it's a common, but it's give you the whole story of my research. It's one figure, is a figure number one, and I try to put those information which is enough to understand what happened in there for the peer-peer relations. Those are working on plant sciences, or those are working on plant genetic modification. So it's a complete story, though they are not interlinked, but it is a sometime you can do this type of figure to attract people and give a single figure to understand your story of transformation and all things, because you use to the legend in here in such a way. So it's a process. Now it's a 1 hour. I wanted to go for the few questions, if then I can demonstrate or can cover other things, but I need some questions. It's very difficult to discuss anything without the options. No, I can hear, or you can put in the chat. Okay, you can put in the chat. Okay, there are questions. So without wasting the time, so it is two different things. What you look doesn't matter. In this thing, you can think that this is this type of figure. This is a smaller one, but when you upload your manuscript figure, when you upload it on the manuscript, then they ask individually, because they need the actual size, and they accept in that way. But you can use this picture for pre-communication like nature and science did. You send your picture in a one PDF, everything put on it. They can go and they can give you a link to submit your full paper. Otherwise, you don't need to put everything right in the beginning. But when you submitted your 10 figures and uploaded in any editorial managing softwares or communicated, you should specify your length or figure. Like what we actually we receive some instruction like the column, length of a column. It's already mentioned. What should be the millimeter size? Either it's inch, either it's centimeter, either it's the millimeter. It's mentioned by the uh, journal. Carefully read the guideline, journal, author's guideline instruction, what actual we should go. So this is the I'm not changing anything, and I prefer, and if another day I have a few paper, those are retracted now, but they manipulate in, they put something which is actually not in that. So it's a dangerous and is a crime now. And this paper published many years back, but recently they retracted because softwares are now are developed, and every paper checked, new paper is obviously checked, and previous one also checked by this software because it's newly updated, and editorial board asks them either retracted or correct the figure. So you need to the figure. And another thing is just how vary the software. Like uh, in my computer, in my computer, if you wanted to know that information, like you can when someone asks you send the original figure, you send this figure and what type of information they can see from here. So you can't change anything in further. So carefully, like which camera you have used. I use Nikon D3200. These all are entrapped in this file, and software to software these are visible. Which which color profile I use, which focal length I used, which the exposure time I used, where I dimension of the figure. So everything is there. That's why sometimes reviewer or editor asks to give the original figure. If they have any doubt on it, they can ask to give the original figure. I'm not using any specific software. I just click to see the info and I get so many information about this figure. So it's very difficult to bypass their eyes because they use more dedicated high throughput softwares on that. Okay, so I stop the screen share here and I go for the question. Okay, there are few questions. Pavitra Joshi. Pavitra now a regular listener of our Bioengine. Thanks, Pavitra, to come every webinar, and I'm sure it's helped you to acquire the more knowledge on it. You put background in pot culture, but how to put background in field experiment? Yes, in field you don't need the background. First of all, when we use the field, actually we try to see all the plants. Okay, that time you don't need to the background. But as I show the crossing material, I took it in a natural condition. So I carry a black paper and chart paper, and with my assistants or with my colleague, I request them to just uh, uh, hold it in the back side of my plant, and I click it. So you can do that, or you can carry a paper or cloth also. In field, cloth is a more suitable because you can wash it, because in field it it should be, it's obvious it get muddy. So you use clothes, for black clothes, not so glossy black. You use a matte black on that, because glossy already reflect the light and it hamper your object. It comes to the experience, or you can ask any photo known persons, though they are not the science, but you don't need their plant science knowledge. You need their camera handling ability. Okay. And for specific pictures, I use the macro lens, because otherwise you can't go for such depth. If you wanted to see the far of insects, then you need a macro lens, which is actually depth. We create the depth. So instrument is very important. Obviously, instrument is very important. Hope you. I solved. Okay, in graphical abstract, this is a very important part nowadays. Okay, then I should go for a screen sharing, because I have 15 minutes on it. What I should start? Graphical abstract. Okay, so first I go for any of my paper which have the graphical abstract. Okay, so I go for this one. Here I came only for the DOI number, because graphical abstract only for the online. You offline very limited papers, so they are printed their graphical abstract. Actually, it's for the online. So this is the graphical abstract. Okay. What is the reason of doing that? Because you can't, you have to get access or purchase this. This is not an open access journal. So it attracts the researcher. You have the two things unique in that online version. One is the highlights. It's not available in the printed version like I have. You can find the highlight here. You find the abstract. You also not find the graphical abstract here. Is a printed one. So in online version, few journals use the graphical abstract. It is optional, but I prefer always give the graphical abstract if there are the option to giving the graphical abstract. Yes. So what is the abstract? So it is the abstract. What you can see appears. It's very short. It's give you the information of whole paper. I use a needle, because in jute is very difficult to transformation. Is a high mucilage crops, and flax is another bast fiber crops. Jute is the highest, most important bast fiber. It's the second one. Jute is the second important crop, fiber crop after the cotton. But in bast, this is the most important. So here I use the piercing. Then I vacuum infiltrate in agrobacterium mediated transform agrobacterium, which has gene of gas. Then I select in kanamycin antibiotic selection. Then I check the histochemical way or in molecular way that gas is a expression or not. But this is the just two experiment. I have many experiment on that, because it's a reputed 4.25 impact factor journal, and you need to give many experiment on that. So if you wanted to see, you can find the actual paper where you find how I clone it. And here I use the same color. As you see that all the promoter is the same color. Okay, to give information. Here I use different type of tone to understand that which one the more important than the others one. You can use this type of picture also to give the information. So it's had so many pictures and figures. So it's a long way, and my plants actually produce the phloem specific expression. Okay, so you have to select the pictures first, how way you wanted to go in your presentation. So graphical abstract basically attracts the people on the paper. Otherwise, no one download or get access. It nearly $40 of the cost of this paper. So it is also very important for point of view of Elseviers. They try to sell this paper as much as possible, and we guys are try to circulate it as much as possible, because it's help to our recognition and also the field of jute transformation and jute science. Okay. Next question is, what is different? Okay, JPEG and PNG. Basically, these are the technical term. I'm not know so many difference, but I know that yes, you can Google it. But if it appeared in my question, then I should try. Is a PNG is more depth for it actually two things I know about the PNG. It incorporated other information too, like file size, file information, background color, font ground color, other information, which is JPG not saved. That's why PNG file size is larger than the JPG of a same picture. So it's more depth images. PNG is preferable than JPG. Why always we have saved photo in JPG only? No, it's not true. JPG because of small size, but I'm not always use JPG. Actually, these are the these are the common settings for JPG, like TIFF. You can send a TIFF photo in your WhatsApp messages. It's not visible. It shows the file, not the picture. JPG is acceptable, and JPG is a very lower size in size. So that's why we use JPG. But I always use PNG or TIFF, not JPG. When I created these pictures, when I create the picture, sometimes JPG is more acceptable, like Google Docs. In Google Docs, TIFF is not acceptable. If you want to paste something in Google Docs, it has to be PNG or JPG. That's why JPG is more common. But in case of paper publication, JPG, though there is an option, you should prefer the TIFF file. In Photoshop, if Photoshop is not available, then no, not necessary you have to Photoshop. There are many software like Adobe Illustrators also there. You have to find anything which can assure the size of your picture. Okay, you can online tool are also available. Picasa also use the size, but you need to online tool or offline tool or whatever which can assure the size. You should not use your picture adjust into the MS Word for the sizing, that you look that okay, it is a A4, it is A4. It's not. It's only for your computer. Your software is look like A4, but it's not a A4 size picture. So don't use this type. You have to need something for high impact or for mainstream journal. Otherwise, you can paste in the Word. They don't ask you the submit to other. What is the option regarding background of the image and photo? Opinion. Okay. In opinion, my opinion is, if your sample or your target sample is visible naturally, then don't bother about the background. If it's visible naturally, but if it's not visible in naturally, then it's very difficult, because you need to you have to use something to create the contrast issue, as I mentioned in slide presentation, that your focus is that this is the role or it's the model, or you wanted to see all viewers to see these things, not these that time you have to create something. You can blur, but it's not a good option. That's why I'm asking, don't use the mobile camera. If you use the mobile camera, there are 2-3 focal length and focal camera. They automatically blur the background, but it's not acceptable, or it should not use for manuscript preparation. So use your camera. But yes, sometimes it's not possible. I'm go to the field and I suddenly see a egg mass. So I should take a photo at least that, okay, wait, if it is known to me or it's unknown to me. So it's not always possible to carry DSLR in everywhere. So you should use the photo in such purposes, but it's not give you the quality of your paper. And another thing I wanted to incorporate, sometime editor asks you give the original picture for their cover photo. Okay, if someone asks you the cover photo, you send your mobile click picture, it's not so good, they reject it. And it's so your every picture should be perfect, and you click in within the mindset that it going to be published. Okay, this mindset is also very important, that it is my final take if it is possible. I have lots of figure and my computer that I don't have any movies or anything, which I have only files and figures in 256. 215 GB is the figures, and so it's required, and I'm always focus my figure. You can see my publication also, all are glossy and attractive figures, my point of view, maybe not from yours, but I'm sure it's you will like it. uh, then you put, okay, what is the difference between is okay, what it's I covered. If Photoshop not these are the same question, why you paste same question. uh, why your opinion regarding how use non-scale to compare two image, how to use nanoscale to compare two image. Okay, first of all, you have to give something within the figure, like in when we took the picture in a microscope, we put a put a uh, put a bar to visualize that the size of the figure. So you need something which is comparable. uh, if you don't have these things, you can put a coin, 1 rupee, 2 rupee coin, or you can put a dollar, or you can put a matches or matchstick on in beside the your object to understand for you, for the further processing. Otherwise very difficult. And then you can use Photoshop also to calculate the comparison. There is a tool, I'm not going for this one, but there are tool which can calculate and compare, but it has to be same. If you not use the same intensity or same zoom in, it will change the, and that time it's not very good way. If you extract some data from it, it always be a error chance. There is a lot of error on doing that in way. uh, there is a question that sir, scaling should be from the base of the pot or from the region of shoot growth. Yes, sometimes because uh, how I use this scale, I have to paste it in a wall or put it in a base. So this is not a matter, because we don't search the scaling when actual figure is prepared. It's very difficult to see the scale, either it is 5 foot or 4 foot or inches, it's not visible, but it gives the visual comparison. Okay, this is the scale and this is the actual. And if someone has interest on it, they can crop this section and can understand. They can crop from the base top of the soil to the height of the plant, or they can subtract the level of the pot size. For me, it is a 10 centimeter. Which type of picture should be put on the thesis? In thesis, JPG is a good. Don't need to TIFF or or PNG. JPG for thesis purpose, JPG is okay, but prefer TIFF file, because sometimes we need to write some review from our thesis. Then you need all the editing once again. That's why I'm using any same pattern for every picture, because I need it for another purposes too. I wrote a review paper. Then I think, okay, we can publish a book chapter using this figure. Okay, and it's common. It's common to common like okay. I go for the skill sharing option. Like as you guys just seen the figure. Now I'm using this figure to write this book chapter. Okay, so here the same plants are there, which is, but orientation is different way I presented here, and I can do that because it's my own paper and I refer this journal also. So it's not a plagiarism, is a different way of representation. So this figure already published, because okay, if you remember, I saw it is a actual published in a book chapter. Actually it is this is a frontier paper on fungus resistant and herbicide resistant jute plant. So you can see that picture. Okay, this picture and this picture is the same. Okay, so another, this picture and this picture is the same. So I use this picture and I refer the journal. But if you have the same picture for further use, you can use in multiple purposes without further editing, because sometimes you may lost the previous one, and sometimes you can lost your actual picture. That's why I also suggest try to upload it in a drive, or try to download from your computer or camera to the external hard disk. And I do another thing. I just buy a 16 GB or 32 GB chip, memory chip. I used when it's full. I just keep it and buy another and plug it. So it's also keep my research elements in chip wise, that 1, 2, 3, and nowadays very low cost of any 32 GB or 16 GB chip, memory chip. So when I fill it, I just replaced it and keep it for my saving for further use. So it's a process. You can do another recycling the chip, which is very common, and collect the data in your computer, upload in the drive. But what happens when you upload in drive, this is not the actual size. It's automatically shrinks some way and uploaded it. When you send some email, you can get an option that actual site or just normal way. In actual site is the site. So 15 GB or 30 GB is very common for the drive, but it's not enough for your photos. You either need so many other types, or you can buy a external hard disk 1 TB, 2 TB and keep it. Okay, there are no more questions, and I'm on time. It's time to say goodbye. Okay, if everything is okay, or there are no questions. Okay, okay, okay. No. It happened to our discussion section because many viewers have subscribe our channel. When we are go for the live, everyone got the messages and everyone thinking this is the webinar. It's not a webinar. I'm not giving any speech. I'm just helping you guys. So it's a discussion section. We don't give the certificate for that. No one give the certificate for that in world. So that's why sorry for the certificate, but I like if you guys have any questions or you can. Okay, so I go for the actual. Okay. Thank you. Thanks. There are a lot of questions maybe in your mind, but this is the very basic. Today, I'm going for the very basic way. Maybe I ask you guys to whether I made a sequel on color and others, font, color, symbols, these things, or just it's enough for understanding. So you guys can ask and suggest if you need such extension about the other topic in manuscript, then I can go. And specifically, I'm not cover all the things, technical things in Photoshop, because I have a plan. uh, like uh, like Akas have the experience on other software, or any others suitable person who have the knowledge of illustrators, Photoshop professionally. Then I request them to give more suggestion, which also may be helpful for me. But it's not a photo editing, or it's not editing some birthday images or marriage images. You edit a serious things. It is the data, so you can't go for everything. Okay, as you go for any image editing, the birthday cake and others. Uh, okay, so uh, it's very. I'm very happy you guys have come here to see. Next plan, I'm planning to another, and I send you to ask you that whether you guys are need some type of training or something. But yes, you don't it is not for the expert one. I'm not covering anything which is for the expert. Because we are created discussion sections for beginners, basically for MSc, early PhD and BSc students. And those are the new and wanted to or revise the knowledge. That is the reason I created, and we think the discussion should be go on. But it don't expect much higher, because it's not possible in a one session. But obviously sequels make you more confident and more uh, more expert in this way. Just keep in mind how to design your figure, how which things you should obey. First, go for the publication, the author's guideline for the publication is sometimes varies. And yes, and tables, and yes, these also are visuals item. We definitely in future we cover the table, what is the meaning of 3-line tables and all, which is required. Figure legend, table legend, anything, because we're planning in such a way, in little bit slowly. So thank you, and sorry there is no password, because you don't need any password, because there are no application form for the certificates. Your presence is appreciated. Thank you. Thanks. No, next discussion I'm not finalized yet. I have a few plan, few expert also. I communicate with them, and I just wanted to establish that process. That's why I did 1 or 2 discussion to establish the bioengine discussion session. Now I introduce other speaker, and I invite to other speakers. That is the reason I start in taking the early classes or early discussions. Okay, so when you go for the video, you can this video is available on our channel. It's also will be available in our website by tonight. The link will be available in our website. And next webinar is on 24th. Okay, from the Ricardo Olivia from UD. So please register, and only registered participant get the feedback link for the certificate collections. It starts at 11 a.m. Indian Standard Time. Thank you. Thank you, everyone. I'm going to end this session. Okay, someone asked that, sir, can we get some training session regarding some skill in biotechnology? I don't know for now, but I don't know. But I hopeful in future. But sometimes it's very difficult. A session of yes, your query is a good idea is good, but okay. Thank you, thank you, thank you. Yes, I plan a plagiarism checking and discussion. Maybe the next talk will be that. Let's see. I have the I have I it is in my mind. I have a planning on that. Okay, so no password for today. Okay, okay. Bye, bye everyone, and thanks Akash and Shoma for assisting me to help me for this session to collect the questions, because it's very difficult single-handedly for doing everything. Thank you. Now I'm going to end